Hello, everyone, and thank you for joining us for today's webinar, Accelerating mAb Drug Development to Meet the Increase in Recent mAb FDA Approvals. I'm Miles Skoda, editor for Life Science Connect, and again, thanks for joining us. So the number of monoclonal antibody drugs making it to market will continue to rise due to the increase seen in FDA approvals of mAb drugs with specific therapeutic indications. To accelerate the delivery of life-saving therapeutics, it is essential to evaluate ways to maximize efficiencies while minimizing risk to stay up to speed with the growing demand. With that said, we've brought in Dr. Scott Jacobia, Director of R&D for Gibco PD Express Services, and Dr. Sanjoy Mukherjee, Principal Application Specialist for Cell Culture and Bioproduction, both from Thermo Fisher Scientific, to discuss this topic. And so they're going to talk about the common challenges and obstacles faced, optimizing development processes, reducing time to market, and selecting the right partner. And we'll be holding a live Q&A session following this presentation. Feel free to submit questions as we go along. We'll get to as many as possible during the Q&A, and any that we can't get to, we'll follow up on after the event. And just a couple quick housekeeping notes. If you run into any issues, please try refreshing your browser first. If that doesn't work, submit a question via the Q&A, and we'll help get you back on track. Finally, the webinar will be available for on-demand viewing after about 24 hours. Just use the same link that you used today. With all that said, I'm going to turn it over to Scott to get us started. Scott?
All right, first I'd like to start by thanking everybody for joining us today, as Sanjoy and I are going to be talking about different topics involved in monoclonal antibody drug development and commercialization. First, I just want to give you a little bit of detail about Thermo Fisher Scientific. Thermo Fisher is a world leader in serving science. We have over 90,000 employees globally, 5,700 of those being scientists and engineers. We invest over $1 billion a year into R&D, and the company's revenue is greater than $35 billion a year. We take a lot of pride in our mission as we enable our customers to make the world healthier, cleaner, and safer. Today's agenda, I'll be talking about some of the past, present, and future of monoclonal antibody manufacturing. I will then be turning things over to Sanjoy to talk about early-stage monoclonal antibody optimizations. We'll both talk about late-stage mAb optimizations, and then I'll be finishing up talking about some commercialization and summarizing the talk and starting our question and answer session. So approximately 35 years ago, monoclonal antibody biotherapeutics gained their first FDA approval. And it took 8 years later for the second mAb to be approved. And after 20 years, the FDA had only gotten to approve its 50th monoclonal antibody. But right about 2015, there was a huge boom in approvals for monoclonal antibodies, where over the next 6 years, another 50 were approved by the FDA to reach a total of 100. So this huge increase in monoclonal antibody approvals per year happened, and now there's currently about 870 monoclonal antibodies in clinical development. So monoclonal antibody biotherapeutics account for about a fifth of the FDA's new drug approvals every year, where cancer has really dominated the mAb biotherapeutic space. Part of this is driven by the Chinese market, as well as manufacturing of antibodies to combat our current pandemic, which is the SARS-CoV-2. So where are we going from here, right? Next-gen antibody engineering, right? So in addition to, manufacturers making full-length monoclonal antibodies, they're also looking at antibody products such as conjugates, fragments, antibody-like proteins. And in addition to these, there is also a huge focus on the biosimilar market, which is currently worth about $4.3 billion, but is really expected to grow to about $8 million by the year 2026. As new targets are identified and the market grows, it will be essential for manufacturers to increase their efficiency to meet this increase in demand for new biotherapeutics. Moreover, becoming more efficient will help accelerate the delivery of these therapies into the market. Okay? And so right now, I think we have a poll, so I'm going to pass things over to Miles.
Great. Thanks, Scott. Yep. So we've actually got 2 poll questions, and we should be pretty quick with these. Hoping you guys can respond to everything here. We're going to use the data from this to kind of craft our webinar strategy kind of going forward and, kind of the information that you guys want to hear from, and also to kind of craft this presentation as we keep going. So the first one, which should be on your screen already, what is the biggest challenge you're currently facing as a mAb developer or manufacturer? And note you can select multiple options here. So insufficient titers, low protein quality, difficulty matching innovator drugs, or raw material issues such as trace elements, supply issues, or serum problems. And I'm going to give everybody about 5 or 10 seconds real quick here just to respond to this one, and then we're going to move on to the next one real quick. All right, that looks great. Lots of answers coming in for that one. So now I'm going to push out our next one. What do you think is the best method for optimizing mAb processes? And again, here you can select multiple options. So switching to an advanced medium, receiving process development support, using advanced analytics, or building a more resilient supply chain for critical raw materials. And again, thanks for your answers to these questions. I'll give you about 5 more seconds here, and then we're going to hand it back to Scott to kind of get the presentation going. All right, and if you didn't get a question or an answer into the question, those should be up on your screen still, so we do appreciate your responses. And with that, Scott, it's back to you.
So some of the challenges that manufacturers face is insufficient titer, raw material issues, and problematic protein quality, especially when you are including biosimilar concerns. And to overcome these challenges, it's important to partner with a global supplier who has commercially available products to support high titer, ensure supply assurance, and that can offer customer services to support custom media development and analytical analyses. I'll now pass the presentation over to Sanjoy to talk about how Thermo Fisher Scientific can help optimize your manufacturing process.
Thanks, Scott. So I will go over the early stage optimization. So media development and optimization is critical to improve productivity and simplify workflows by increasing titers. This would be to customize your in-house formulations or development of advanced catalog formulations, which are AOF and CD, available in multiple different formats and optimized for different cell lines. To name a few, the Gipco Dynamis media, which is designed to offer the highest batch and fed batch performance, the Gipco High Intensity Perfusion CHO media, or the HIP CHO, designed to enable high titers regardless of the perfusion platform, and our newly launched Efficient Pro media, which is an innovative CHO media that delivers high levels of specific productivity as well as volumetric productivity. Going forward, of course, proper supplementation with feeds, reagents, and additives to your process can reduce variability. It also improves titers and optimize protein quality. Some of the innovative Gipco feeds and reagents available are the newly launched Efficient Pro feeds, which are CD, and a whole range of Gipco peptones. We also have the Glycan Tune supplement to achieve high titers and improve protein quality. everything starts with the cell line. So a stable cell line development is a crucial first step towards achieving high titers in a short time and to achieve a stable process. Convenient, easy-to-use kits like the GIPCO Freedom Cell Line Development Kits contain all the necessary components that can fulfill needs of high titer, short timelines, regulatory friendly, and low licensing cost. Whether batch, fed batch, or perfusion process that works for your MAB manufacturing, GIPCO has media and supplements that fulfill your needs. The GIPCO HIPCHO media is a AOF CD media for intensified fed batch, concentrated fed batch, N-1, as well as continuous perfusion. The Function Max Titer Enhancer increases the efficiency by improving specific productivity. GIPCO Rapid Prototyping Service is a fast pilot-scale media manufacturing service where fast turnaround times of 10 to 20 working days and 200-liter or 10 kg batches can help in your early-stage MAP development, helping to kickstart your products. Some of the benefits include testing the manufacturability in pin mill and FITS mill, as well as conversion in formats from liquid, dry powder, and AGT prior to cGMP scale-up. GIPCO PD Express Services also provides advanced analytics, which is key in supporting process development and helps in troubleshooting and optimization to boost yields and improve quality. Analytics such as spent media analysis looks at addressing nutrient needs, and KDI, or key driver identification, can be used to determine components which is driving performance. This is an upcoming technology which is known as the multiomics. It is opening new doors in pharmaceutical process development, application of proteomics and metabolomics to identify intracellular pathways and to establish how media components are used by cells is a revolutionary new way to optimize media. GIPCO PD Express service includes the multiomics capabilities, which can define the roadmap for the cells by learning how media optimization can go forward and proteins are expressed. An ideal partner such as Thermo Fisher will support your unique early-stage cGMP sourcing needs and provide access to a broad portfolio of cGMP chemicals from reputable suppliers to help you mitigate risks, increase productivity, and reduce your total cost of ownership. I will now pass it back to Scott to talk about late-stage optimization.
Once you've been able to identify your optimal basal and feed media, it's important to consider the medium format that will best fit your process. Some factors for consideration are your in-house capacity and time for media preparation, your availability for in-house storage space, shipping costs, how much of the media that you need, and just overall costs associated with shipping, storing, and preparing the media formulation in-house. And so an ideal supplier would offer you not only high-quality products, but also the suitable batch sizes and packaging and formats for your particular manufacturing process. Thermo Fisher offers multiple formats, including 1X liquid, dry powdered medium, or DPM, and our advanced granulation technology known as AGT. The liquid and DPM formats obviously have pros and cons, as you can see on the slide, where the dry format is more economical for shipping and storage, has longer shelf life, but there is reconstitution, can be labor intensive and costly, and the liquid, which is easy to use and has a reduced risk of cross-contamination, can be expensive to ship, especially across oceans, and has a shorter shelf life. The AGT format leverages the pros from both the liquid and from the DPM, and it's able to be used, and it is an easy-to-use, pre-adjusted granular form that delivers all the benefits of the liquid media without the cost, storage, and transportation issues. At Thermo Fisher Scientific, we can work with you to help develop a custom formulation specific for your platform through our GIBCO PD Express services team. We can manufacture your proprietary formulation in your desired format, and we're able to work with you to make variants of existing formulations that you might need through our rapid prototype meeting manufacturing group in an effort to help your process development team. I'm now going to turn it back over to Sanjoy to talk about the next section.
Thanks, Scott. The bioreactors are an important piece of equipment and essential in supporting the late-stage production of MAP products. What we show here is the high performer Dyna-Drive single-use bioreactor, which is our latest advancement in sub-technology. It offers better performance and is scalable from 50 liters to 5,000 liters with a 20-to-1 turn-down ratio at higher scales. Challenges associated with scale-up can be overcome by solutions such as digitalization. Some of the solutions includes improved efficiencies with single automation across both hardware and software platforms, and streamlined, secure, traceable data transfer with fully integrated solutions in an open architecture system. Thermo Scientific TrueBio software powered by Delta V control systems from Immersion provides a simplified and affordable solution to meet the needs. It delivers a unified experience between controllers when transferring processes through the various stages of drug development. Whether you are a CDMO, large biopharma, or a small startup, this software can optimize your MAB manufacturing. So we have a good support team, which is another benefit of working with Thermo Fisher Scientific. And we have dedicated team of scientists, regulatory specialists, business development, and project management to help shorten time to market and reduce cost. I will now pass it over to Scott to talk about optimizing commercialization.
It's essential to choose a partner with expertise and experience throughout the commercialization process. And we offer expertise in all of these following areas: process characterization, advanced analytics, product development, process optimization, tech transfer, biotech business modeling, life science investment evaluation, project risk mitigation strategies, timeline and management, CDMO and media manufacturing support, primary and alternative cGMP chemically sourcing strategies, and process liquid buffer preparation process simplification. Additionally, your partner should have large strategically placed manufacturing footprints that are capable of supporting redundant manufacturing of both animal origin and animal origin-free products, as well as having world-class logistics and distribution solutions. This slide shows the facilities that support media, reagents, and serum manufacturing, as well as our Gibco PD Express service offerings. And you can see that we have a global footprint for this our manufacturing network. Having global manufacturing sites allows us for timely supply by having regional custom solutions for distribution and stocking to help meet our customers' needs, all while maintaining high quality assurance. So in summary, hopefully we've been able to adequately communicate three major points that are critical for establishing a robust monoclonal antibody biotherapeutic manufacturing process. The first being the importance of optimizing your MAB process and the considerations that go along with that process, such as media formulations, cell line development kits, and rapid prototyping. The second being the importance of media formats and bioprocessing equipment. And finally, optimizing processes to ensure supply during commercialization. Thermo Fisher Scientific is here to support you through your manufacturing journey. So now we would like to take any questions you may have.
All right, awesome. Sanjoy and Scott, a wonderful presentation, lots of great information. Now we're going to get moving into the Q&A. So we've got a good amount of questions in already. Again, if you have any questions, submit them through the top right corner of your screen. There should be a little Q&A module, and that'll let you kind of access the Q&A and turn everything on there. So let's get started. I've got one here. Roughly how many molecules can we monitor using multi-omics workflow? How is it different than the traditional media development?
So the traditional media development method, which is based on spent media analysis, it can monitor 50 molecules, while multi-omics can monitor around 8,000 molecules, which allows us to identify specific pathways and tighter influencing pathways. Thank you. Thanks for the question.
Yeah, awesome. Let's see. I'm just trying to look through this list real quick. So which are the MABs that have gained FDA approvals?
So as I mentioned earlier in the talk, that there's actually been over 100 that have been approved, and there's actually even more in review. I don't have a list of them handy, but a quick Google search can give you that list. But just to name some of the top hitters historically, you have Humira, you have Rituxan, you have Herceptin, Remicade, as well as some additional monoclonal antibodies that are actually in review again to combat SARS. The coronavirus.
Okay, perfect. Let's see. So someone said, I'm interested in getting rapid prototyping. That's all they left it at. But maybe you could tell us how they could get information about that, or request a quote.
Sure. The best way to do that is to actually contact your Thermo Fisher sales representative, and they can give you tons of information on our Gibco Rapid Prototyping, again, which is a non-GMP pilot media manufacturing facility. And working with your sales rep, you should be able to get all the information that you would need to get a quote for whatever formulation you want created.
Okay, great. Let's see. So this one here says they're looking to leverage a partner's scientific knowledge to help make informed decision in partnering. So I guess how can they leverage Thermo Fisher's scientific knowledge to help them make an informed decision in partnering?
So there's a couple different ways. So firstly, if you're able to identify your own pain points, you can have a discussion with either your sales rep or your account manager, who then can come in and bring in either our TSSs, who are technical sales specialists, to discuss how our catalog products might be able to fit into your workflow. If you need a higher level of expertise, maybe you don't fully appreciate or understand your pain points, then they may bring in one of our FAS team members, which are the field application scientists. And these folks really will be able to help not only with how our catalog products may fit into your workflow, but also can initiate discussions on custom product development to increase the robustness of your process, and also bring in our R&D teams where needed, because they can be brought in for maybe harder problems, as well as our manufacturing sciences groups, our program management teams, to help with, supply options, mitigation of, some of the downstream manufacturing processes.
Thanks, Scott. That was a great answer. So let's see. I've got a long one here. So if you need anything repeated or anything, let me know. They are asking, we are a CDMO developing fed batch process in CHOK1 cells and have challenges with specific productivity and charge variants. Will Efficient Pro Media and Efficient Pro Feeds help me to overcome these challenges? And what would be your recommendations for these type of cells? They're saying they've tried medias and feeds for most vendors, but the problem still exists.
Appreciate your question. So as I've actually presented, we have launched the Efficient Pro Media and Feeds in December of last year, because we got VOC from the market saying that, they have challenges in regards to the specific productivity as well as the glycosylation pattern, and can they have just one feed? So Thermo Fisher Scientific has developed a media and a feed for CHOK1 cell lines, having those specific interests in mind, looking into specific productivity as well as the charge variant analysis. And the one that I would recommend here is the Efficient Pro Feed 1, which works well with the Efficient Pro Media for CHOK1 cells. For the CHO-S cells, the Feed 2 works well for CHO-S as well as DG44 cell lines. Thanks for the question.
Awesome. Thanks, Sanjoy. Let's see. So we've got a few more in here. Again, if you have any questions, top right corner, submit your questions. We'll definitely get them answered. I've got, let's see. So how do you train researchers to understand the PD and manufacturing struggles, like chemistry choices or equipment choices, and the impact on the speed to market?
Well, so internally, we have built our FAS team, our field application scientist team, from folks that have actually worked at our customers. So they have had experience bringing a biotherapeutic to market. So they understand some of those pain points already, and using that in combination with our FAS teams, our R&D team, and our manufacturing sciences teams, developing great working relationships with our existing customers has allowed us to really gain insight into what their processes are and how their pain points come up, and that being able to use, and understand their pain points that our customers may have allow us to innovate proactively on developing, new products or new processes to help them, almost before they have the problem, because, we're constantly trying to use voice of customer in our product development, such that we can solve a problem before they necessarily realize that problem at their site.
That's perfect. Thanks, Scott. Let's see. Another one here. What what is your experience with media design and optimization for microbial pDNA manufacturing?
So I'll start this one, and I'll see, Sanjoy, if you have any additional comments on it. But Thermo Fisher has launched a catalog product for microbial cell growth. And though we don't necessarily have experience with pDNA manufacturing, we do have, as was mentioned in the presentation, our GIBCO PD Express Services team, which has the expertise, and we have worked on microbial media development projects. And so, again, depending on exactly what you need, we can design a custom service project to help increase productivity of your system. I don't know, Sanjoy, if you had anything to add into that, or?
Yeah, thanks, Scott. So some of the things that work great in the microbial systems are the peptones. And as you know, Thermo Fisher is the market leader for peptones. We start looking into peptones as well, how to complement or some of the peptones that work great in the microbial environment. We have a service called KDI, a service which is the key driver identification, looking on certain peptones, certain components in the peptones, and that work great in the microbial space as well. We will utilize that knowledge in this media design and optimization.
Okay, great. So another one here from early on in the presentation. The question is, in slide 7, you mentioned that the Chinese market was driving the direction of mAb manufacturing. Can you talk more about that and kind of what is the mechanism by which this is happening?
Yeah, and maybe I can clarify exactly what I was trying to get across is the overall market of mAb manufacturing is growing at a very high rate within the Chinese market. And so, the direction that they're driving it is really just that we need to focus more time and energy on the innovators within the Chinese market to make sure that we're able to support them through supply so they could get their biotherapeutics out to the market.
Okay, perfect. Thanks, Scott. Let's see. So I've got one more here. I'm not sure if you've got this data on hand, but could you talk about how many MABs Thermo Fisher has been involved with through approval?
Well, so Sanjoy and I are more on the upstream R&D side, and I actually don't follow through to see which ones that we've actually worked with have gained approval, as that would be more like a business development type. So I don't have an answer for that. I'm not sure, Sanjoy, if you would be able to answer that either.
Well, being in the market and one of the leading players in the market, what I would say is it's thought we don't know exactly that number, but we are a leading player. And as you know, more than 100 antibodies are in the market. We are a leading player in that space. So I don't have the exact numbers.
Yeah, no, that makes perfect sense. And again, to whoever asked that question, if you were looking for specifics, I'm sure you can reach out to Scott or Sanjoy. They can get you or at least put you in contact with the person to help gather the data to share. So definitely not a problem. I know you guys would have that data, but of course, like you said, you're not involved specifically. So that's great. I think with that, we're going to wrap up here. Sanjoy, Scott, thank you so much for the great presentation, for going through all these questions with us. If you did have any more questions, submit them through the Q&A. Reach out to any of us here, and we'll get you connected to the right person. Thank you to the audience. Thank you all for joining us today, and we hope you all have a great rest of your day, and we will see you next time. Bye, everybody.
Thanks a lot.
Bye now.
Bye-bye.